So our battlefield has from one side the phagocytic immune cells that are trying to kill the pathogen, the invading pathogen, and the parasites use different strategies such as generation of reactive oxygen species and cationic fluxes to kill this bacteria, the gum called Candida albicans. So, and from another side, we have Candida stress responses that ensure its survival. So basically, Candida is resistant to these stresses and it survives. So now the question is then, how does phagocyte can still kill the pathogen if Candida is so resistant to most of these stresses? And the answer is here. What was found using fact analysis that combination of cationic and oxidative stress, even in vitro, can kill Candida albicans much more potently than the corresponding single stresses. And it was like in real life happening because most of the studies looked only for the survival of the pathogen using single stress, but we had the effect is not cumulative, and it's really interestingly. So then we had a look further, and we found that microarray datasets suggest that most of the genes that are upregulated following oxidative stress treatment are not really silent in response to the combinatorial stress treatment. So, and then the logical idea was to look on the regulation. So, majority of these genes are regulated by CAP1, this epi-like transcription factor in Candida albicans. And we also proved this fact because most of the members from CAP transcriptome are not induced, like our northern analysis suggests, in response to the combinatorial stress treatment. And it was really interestingly. So then we had a look on CAP regulation. In normal conditions, under non-stress conditions, CAP1 is reduced, is floating freely in the cytoplasm, and it can go to the nucleus. But in the nucleus, we have cream exporter that can bind to nuclear export sequence in the CAP protein, and CAP1 is like going back to the cytoplasm. So we have no antioxidant enzymes expression, and we have like normal non-stress conditions. And I was really like looking and realizing that my favorite protein basically doing like a moonwalking movement where it's trying to go to the nucleus, but here we have cream one, and due to this complex appearance, it has to go back. So basically, it's not a real situation. Then what's happening when we have say hydrogen peroxide stress? CAP1 is it's oxidized on conserved cysteine residues into the cell, so it can still go to the nucleus, but it cannot bind to CRIM1 because its nuclear export sequence is blocked. It's somehow because of folding of the protein is changed. So, this mechanism of CAP1 accumulation in the nucleus allows it to trigger the antioxidant enzymes expression. So, and as a result, we have the Candida cell survival. And then we were interested what then changed when we add the salt. What's the difference? So the first thing we looked in the CAP1 oxidation profile. So we use this alkylating agent called AMS that can specifically bind to reduce cysteines and change the mobility on your Western blot gel, on your protein gel. And what we found that during combinatorial stress treatment, CAP1 is forming really heavy form, which is we suggested it hyperoxidized form, but this form is transient and it disappears after 1 hour post-stress stimuli. Also, we looked at that phosphorylation profile of the protein, phosphorylation of CAP1 is delayed in response to combinatorial stress treatment, and it's consistent with delayed nuclear localization of CAP-GFP, and it's consistent with the delay of CAP-dependent gene expression. So it was interesting. We thought, yeah, we found the explanations of this, but nothing is so simple. Then we were wondering whether if we got the overoxidized form of CAP1, so it might be dependent on increased ROS level. So we had a look on the ROS level inside of the cell, and we found that this combinatorial stress treatment triggers at much more higher ROS levels inside of the cell. So we thought that it might explain the CAP overoxidation. And so we decided, why don't we just use very high physiologically non-relevant doses of hydrogen peroxide, treat our cells, and have a look whether we can get the same non-active hyperoxidized cup form. And basically we succeeded, because as you see with increasing H2O2 concentration, our cup one is hyperoxidized, and if you treat the cells with 10 millimolar peroxide, it's almost trapped with this inactive hyperoxidized form, and it's inactive. And it's consistent with the delayed phosphorylation and with the delayed cup-dependent gene expression. But what's interestingly is that it cannot explain our mechanism fully, because this delay it's really like maintained only not till 1 hour, but only 10 minutes. So the second mechanism that we found was the interaction of cup with cream monkey. So we made it cool down, and we looked at really under stress conditions with hydrogen peroxide alone, we cannot bind to cup one. We have these puzzles, protein puzzles that don't match each other. We have couple accumulation. But when we add the salt to the mix, this changes the structure, and the salt somehow promotes this cup-cream interaction. So cup can't accumulate in the nucleus, and as a result, to sum up, this other combinatorial stress-induced cup inactivation mechanism. From one side we have cup overoxidation, from another side we have increased interaction and salt-induced increased interaction with cream exporter, and we suggest that such a mechanism underlines the potency of neutrophils to kill the pathogen in healthy hosts, which leads to the pathogen death. Thank you very much for your attention. I thanks to all my team from Newcastle University, to my brilliant supervisor John Quinn, to our partners from Aberdeen Fungal Group, from University of Exeter, and for funding NHS, and for all of you for your attention. Thank you very much.